Szanowni Państwo, jest godzina 13:00. Witam Państwa na kolejnym spotkaniu Nowe oblicza biochemii. Będziemy transmitować dzisiaj wykład pani dr Karoliny Mikulskiej-Rumińskiej. Wykład będzie w języku angielskim. Na zakończenie, po wykładzie, będzie można zadawać pytania w trybie Q&A, czyli w postaci czatu. Pytania można napisać i pani Karolina odpowie na pytania. Zanim poproszę panią Karolinę o wygłoszenie wykładu, kilka słów o karierze pani Karoliny w ostatnich latach. Pani dr Karolina Mikulska-Rumińska jest absolwentką fizyki medycznej na Uniwersytecie Mikołaja Kopernika. Wykorzystuje ona w swoich badaniach narzędzia biofizyki obliczeniowej. Ostatnie 6 lat poświęciła na badanie procesu ferroptozy występującego w chorobach Parkinsona, nowotworach, astmie czy w sepsie. Jej publikacje zawierające przełomowe odkrycia nad zjawiskiem ferroptozy zostały opublikowane m.in. w Cell i w Nature Chemical Biology i są szeroko cytowane na całym świecie. Odbyła 2 międzynarodowe staże naukowe na Politechnice Federalnej w Lozannie oraz na Uniwersytecie w Pittsburghu oraz odbyła także staż w firmie Adamet. Brała udział w wielu grantach, w tym w 8 jako kierownik. Te granty to Preludium, SONATA z Narodowego Centrum Nauki. Jest beneficjentką wielu stypendiów, m.in. tegorocznej edycji L'Oréal-UNESCO Dla Kobiet i Nauki w kategorii habilitacyjnej. Jest polską kandydatką do międzynarodowej nagrody L'Oréal-UNESCO International Rising Talents. Ostatnio została wymieniona na liście 100 najbardziej inspirujących Polek w roku 2021 według Forbes Women. I tak w największym skrócie wygląda życiorys naukowy pani dr Karoliny Mikulskiej-Rumińskiej. Pani Karolino, poproszę panią o włączenie mikrofonu i kamery.
Thank you very much. Thank you very much for this nice introduction. It's a pleasure to speak in front of you. I would like to thank organizers for giving me the opportunity and inviting me to give this talk. As you can see, I will be speaking about ferroptosis process. This is a programmed cell death, which was discovered quite recently because it was 10 years ago in 2012. And since then, this process was explored by researchers. So each year we can see many different findings and new correlations with different types of diseases. That's why exploring this topic is very excited, and I'm very happy to do that. The outline of the presentation is following. So I will say a few words about ferroptosis process and how lipoxygenases are involved in it. Then I will briefly explain which methods I was using. I was using mainly computational methods. That's why I will focus on those. However, experimental validation was also provided, and it was provided by experimentalists from the University of Pittsburgh. And then I divided my results into 5 parts, probably the most interesting. Yes, I will speak a little bit about lipoxygenase, PBP1 complex, which is present in humans. And then I will say a few words also about bacterial lipoxygenase. How is it different compared to the human form? I will also speak about ferroptosis inhibition, which we did using nitric oxide and the most common ferroptosis inhibitor. And this is ferrostatin 1. And at the end, I will focus a little bit for a while on the phospholipase E-plato-beta, which we showed that it's able to avert ferroptosis, which was a quite huge discovery for us as well. Cell death may happen in basically 2 ways. It can be accidental cell death when we have an injury, wound, or something will bite us, and our cells are dying in a non-regulated way. But in our organism, we have something which we call regulated cell death. This is because our cells can divide it and replicate a certain number of times, around 50, and after that, they need to be removed from our body, and we need to replace them with a new one. If this is not happening, then they gain immortality, and that leads to cancers. And regulated cell death, you may say that it's integral part of our organism. And we can divide it into two types, whether the membrane remains intact or not. We have apoptosis, the most famous and discovered 50 years ago, or we have several other programmed cell death, such as ferroptosis, peroxidation, necroptosis, and a few more. Those you can see that they were discovered quite recently. Ferroptosis is one of the newest one. And the characteristic feature of ferroptosis is the accumulation of lipid peroxides. So here you can see the membrane, yes? And if we have a membrane and peroxidation is when we have, in the presence of the iron, molecular oxygen will be built into the one of the chain of such lipid. That caused the massive lipid peroxidation to another lipids, and finally membrane damage, which is which leads to the ferroptosis. Ferroptosis is correlated with many different kind of disorders and diseases. Here you can see examples of few of them, so such as sepsis, cancer, or neurodegenerative diseases such as Parkinson's, Alzheimer, or Huntington diseases. It was also present in the degradation of tissue in brain trauma, kidney, or asthma. It is important to explore this mechanism because induction of ferroptosis has a great potential in a cancer therapy, while suppression of this process may be helpful to stop neurodegenerative diseases. Yes, because it has been shown that iron, high level of iron, is present in brain of people who has Alzheimer, Parkinson's diseases. So there is a correlation between ferroptosis and those diseases. As you can see, ferroptosis is quite complex. It involves many different pathways and many different proteins and small molecules, but and many are not explored and not found as yet. So many things are still unknown, but we know that it can be triggered in cells by the loss of the control of 1/3 major processes: lipid metabolism, which is here, lipid peroxidation, and iron and fuel regulation. I was focusing on the lipid peroxidation because it involves lipoxygenases, those enzymes which are subject of my studies. Lipoxygenases looks like this. So here you can see 3D structure from crystallography. And lipoxygenases contain iron in the catalytic site, which is coordinated by 3 histidines and isoleucine. And this enzyme is very important because this enzyme can give the biggest contribution to the generation of those lipid peroxides, which are accumulated in a ferroptosis process. The most common substrates for lipoxygenases are arachidonic acid and linoleic acid. Here you can see an example of 4 types of lipoxygenases. As you can see, they have a digit in the front of the name, and this digit corresponds to the carbon atom, which will be changed, to which this lipoxygenase will build molecular oxygen. And in humans, we have 6 of them. Here you can see 4. Isoform 15-LOX1, isoform number 1 and 2 was the main subject of my studies. As you can see, they can generate different types of lipid peroxides, but they can be also changed into the bioactive lipid mediators, which can be then used into the cell cycle, survival, or migration. Yes, here you can see the computational approach which I was using. I am not sure how familiar you are with those computational tools. That's why I will briefly explain a little bit them. So I was using molecular docking, and we have two types of molecular docking. It can be between two proteins or between protein and ligand. I was using molecular docking to predict the orientation of PBP1 versus lipoxygenase, and also another to predict the orientation of the substrate in the catalytic site. Sorry, because that should be an animation which should change the conformation of the substrate, but for some reason it does not work now. Yes, so with molecular docking, we can predict the most stable complex, yes, between two proteins or protein and some ligand. To describe the behavior of atoms of those complexes and those proteins, I was using two approaches. One was elastic network models, which are quite common, and they were discovered 20 years ago or more. Yes, if we want to describe the movement, the long-time dynamics of such proteins or protein complexes, we can use them. And I was using Gaussian network model and anisotropic network model. I will show a few examples of the results which I was obtaining. And I was using also molecular dynamic simulations, yes, to simulate the behavior of atoms, because we know that proteins are dynamic structures. That's why it's better to describe it in such way than use just rigid docking. And quantum calculations I was using to parameterize the catalytic site, because this catalytic site is non-standard. So I had to obtain the geometry and also partial charges from quantum chemistry to describe it in a way. Molecular dynamic simulations, maybe you are familiar with that. I'm not sure. That's why I would like to explain a few basics. We are simulating the behavior of atoms. Here you can see atoms, which are then also displayed in such form, in a secondary structure form. But with those simulations, we have a ball spring concept. It means that atoms are like balls, which are connected with the springs. And we have a time which is divided into a discrete time steps, so which are normally between 1 and 2 femtoseconds. It depends on the simulation. To obtain the position and velocity of each atom in our simulations, we are using Verlet algorithm and Newton notation of motion. And with those equations, we can obtain for each step position and velocity of our atoms. Forces which are acting on our atoms are calculated from the energy, and energy of atoms are calculated using potential energy functions, which we called force field. Here we can see the main core of the force field. We may have a different type of force fields, such as GROMACS, OPLS, and CHARM, and so on, so many of them. They contain similar core, which contain a bonded term and non-bonded term, yes, but they can also contain some other additional components. However, we should know that if we have more components, we would need more computational resources to compute the simulations. So we need to choose whether we want to have a better description and more expensive simulations, or a little bit less expensive simulations and also a little bit less quality. So I started to work on this topic. I started my journey with ferroptosis in 2016 when I joined to the Yvette Bahar lab in Pittsburgh at the University of Pittsburgh. It was in 2016, so it was 4 years after the discovery of ferroptosis. And after a year of work, we published the first paper in Cell, where we showed that PBP1, PE binding protein 1, this is the full name, PBP1, can bind lipoxygenase number 15, and to be exact, isoform number 1 and 2. It was tested for both from human. And upon this binding, it can switch substrate specificity of this enzyme from arachidonic acid to arachidonyl PE, which we call SAPI. And our experimental colleagues provided many different types of experiments. Here you can see one of them from fluorescence microscopy. So you can see that those two proteins are colocalized in a similar region. Yes, if we combine red and green color, it will give us yellow, which is present here, yes? And those two colors correspond to our proteins. Yes, so this complex, right now, the main substrate for this complex is arachidonyl PE. Yes, so upon binding with PBP1, lipoxygenase changed its substrate specificity from arachidonic acid to this phospholipid. Yes, and after catalytic reaction, we can observe that we have OOH group, which is built at a C15 carbon atom. So this is lipid peroxide, which is generated by this complex. So the complex can trigger ferroptosis, yes, because it can generate those lipid peroxides. And this is what I said here. And what I wanted to explain also, that there is another protein which we call GPX4, and this protein can change lipid peroxide into the lipid alcohol. But if we don't have this protein, then, or we have too many of those, then it will lead to ferroptosis. Yes, so we have some mechanism in our organism which try to protect us against ferroptosis, at least if we have a smaller number of those lipid peroxides. Okay, in the cell paper, we showed also how the complex looks like. So the binding interface between lipoxygenase and PBP1, and we suggested some mutations such as proline to glutamic acid, and also to cut this C-terminal helix of PBP1. And our collaborators checked in experiments that this complex is not formed after those modifications. With anisotropic network models, in which we can see the movement of the proteins, we showed that cooperative movement is gained upon PBP1 binding. So this movement is not reachable by lipoxygenase alone. Yes, it's not available. But if we have a PBP1 bonded to this lipoxygenase, we have completely new mode. And with this mode, if we take this dynamic structure and we will perform molecular docking, so we will check where the substrate will bind, we will see that compared to the static one, to the static structure, we will have completely new binding sites, which are near the catalytic site, which was not available for a just static structure from docking. We also use Gaussian network models. Gaussian network model, with this model, we cannot see 3D movement like with ANM. However, we have a color. So blue color, it's a coupled region. Red is also coupled, and they are moving into the opposite direction. So like this. So yeah, the movement of lipoxygenase is like this, but when we will add into the system PBP1, we will see that this PBP1, this protein, will be coupled with this helix, which can open and close catalytic site for a substrate. And also with beta barrel, which is in the membrane normally. And if we look at the bacterial lipoxygenase, which is a little bit different because it does not contain beta barrel at all. It contains additional helices, which are not present here. And what is interesting, that this bacteria does not contain PBP1 at all in the system. And it also does not contain the substrate, which is favorable by this complex, human complex. But when our colleagues, experimentalists, added SAPI, so arachidonyl PE, into those bacteria, they started to produce the same lipid peroxide, which is generated by human complex. We can see on the GNM profile that the dynamics, yes, this red color correspond to this parts and blue to this parts, that the profile is quite similar. So we are thinking that those additional helices may play a role of PBP1. Yes, and here another nice example of anisotropic network model. Here you can see a passage between open and closed form of bacterial lipoxygenase, maybe again. Yes, and those structures are from crystallography. Excuse me. Okay, so the next topic I would like to speak about is ferroptosis inhibition. Here we can see the catalytic cycle of lipoxygenase, and in it we have a molecular oxygen, which will be built into the one chain of the lipid. And we ask ourselves, what will happen if we will add nitric oxide into the system? And this is what we did, and we published in the last year 2 papers on that subject. In simulations, we considered 2 systems, with the substrate and without it, and also with PBP1 without it. So many different types of simulations with different combinations. And I was checking also how the ratio of different small molecules, nitric oxide and oxygen, will change the dynamics of the complex and so on. And here you will see, yeah, we discovered that if we have nitric oxide in this system, it will start to compete with oxygen for a binding site, whether we will have a substrate in the catalytic site or no. In both cases, they will compete for this binding site. Yes, this is a substrate, and those light balls, those are places where molecular oxygen can build. Yeah, in a few, now you can see the competing between those 2 small molecules. And finally, nitric oxide will stay in the catalytic site and molecular oxygen will go out. Yes, so what we showed in this study is that we had a few entrances into the catalytic site. One, the most common, is entrance 1. Here you can see the pathway into the catalytic site. And molecular oxygen and nitric oxide, they are using the same entry pores to the catalytic site, so we cannot distinguish 2 types of entry pores for one or another. They are using the same. We have entrance 2, which is almost completely covered if we have a PBP1 in the system and substrate. And we have some additional entrances which are rarely used. Here we can see how nitric oxide and oxygen were competing for similar sites and which entrances they were using. Yes, whether it was entrance 1 or entrance 3. Yes, here we can see some details of those entrances. So we check which residues are involved in this binding, which residues direct those small molecules into the catalytic site, and which residues are stabilizing the substrate in the catalytic site. Yes, this is the entrance 1, which is the most common. First, nitric oxide or oxygen was binding here, and then it could take 2 places, S1 or S2. Yes, and this is what you could see on the movie when nitric oxide was here and here was oxygen and it was trying to get in here. Yes, and from entrance 3, we can see that we have common binding site for S2. Yes, this is the same place for both entrances. Yes, with experiments, my collaborators also showed that nitric oxide will compete with oxygen. If we add nitric oxide into the complex, we will see that peroxidized product will drop. yes, the number of it. What was interesting also in the lipidomics experiments was that if we have a complex between lipoxygenase and PBP1, we are going to have more nitrosylated product. Yes, so that will be product with NO instead of oxygen. Yes, compared to the lipoxygenase alone. So we was wondering what is happening there and how come we may have a different number of products here. And simulations could explain this. If we have a PBP1 in the system, we could observe that both molecules, nitric oxide and oxygen, were able to go into the catalytic site. Yes, blue color corresponds to the nitric oxide and red to the oxygen. And this is substrate here. But when we had lipoxygenase alone, we could see that only oxygen is going inside. And that was, we were puzzled for a while why we couldn't observe any nitric oxide in the catalytic site, because we still have some results, some data that it is generated. So I tested lipoxygenase with arachidonic acid, so the most common substrate for lipoxygenase alone. And I checked that I am obtaining both molecules into the catalytic site. Then I find out that we have a cavity, which is, this region is modified upon PBP1 binding. So this cavity does not exist if we have a PBP1. And normally in this cavity, nitric oxide is accumulating instead of going into the catalytic site. So that was the reason. Yes, upon binding with PBP1, these interactions are not happening actually. So what I did, I increased the number of nitric oxide in my simulations just to check what will happen if you were gonna have more nitric oxide molecules. And then I finally have seen that some molecules are going into the catalytic site, which correspond quite well to the experimental results. Yes, we have a lot of nitrosylated product for PBP1 LOX complex, but much less for lipoxygenase alone. And the last topic I would like to, sorry, not the last, almost the last topic I would like to say about is ferrostatin studies. So we were studying ferrostatin 1. This is the most common ferroptosis inhibitor, but it's not LOX inhibitor. It is a poor LOX inhibitor. So scientists were puzzled why it is weak LOX inhibitor, because LOXs generate lipid peroxides. In this publication, we showed that ferrostatin does not affect lipoxygenase alone, but it will affect the complex. So the missing puzzle was PBP1. It will inhibit the complex, yes? And here I performed another molecular dynamic simulations. It was with lipoxygenase and alone and with a complex with PBP1, just to obtain a different conformations from principal component analysis and mean shift algorithm, which I used. So when I had a different representative conformations, I performed molecular docking of ferrostatin 1 onto two systems to obtain the best positions or initial positions of ferrostatin. And those systems I took to a molecular dynamic simulations to check whether the PBP1 will be able to bind to the lipoxygenase in the presence of those molecules, yes, of ferrostatin. For two of them, I could not obtain, let's say, any useful result. I could observe binding with PBP1. So those two red and green conformations does not affect PBP1 binding, but the blue one, I could see that PBP1 was not able to bind to the lipoxygenase. Only for two conformations I could see binding, and this at the spot which we obtained from molecular docking and confirmed in Cell paper. And however, in those two cases, I could see that ferrostatin changed its conformation, changed its binding spot. So that's why they were able to bind to the lipoxygenase. And without ferrostatin, we could see that PBP1 was binding to this binding spot. And the last topic I would like to say a few words. It's a phospholipase IPLA2 protein, and we showed that it's able to avert ferroptosis. It means that this protein can make a hydrolysis of peroxidized phospholipids, which are generated by the complex of PBP1 and lipoxygenase. And in such way, it's able to eliminate this ferroptotic signal. Yes, and we showed that this protein is a new ferroptosis regulated, so that was also something new. And mutation of this phospholipase may be implicated in Parkinson's disease pathogenesis, because my collaborators, they had samples from patients with Parkinson's disease, and yeah, we could observe, they could observe that this mutation of this phospholipase, yes, it's a characteristic feature there. Okay, so in a computational studies, the first step was to perform simulations with a membrane. Here you can see the composition of the membrane. So it was, I had a different type of lipids. Some of them were non-standard, such as this one lipid peroxide, which is a product of PBP1 LOX complex. This is the brown one, which is here. And I performed molecular dynamic simulations of those membranes. Here you can see example of each lipid in the system. So you can see the dynamics of it. And this is the lipid peroxide. And at the end of this animation, you will see that molecular oxygen is going into the surface. Yes, so so in these studies, we showed that in this lipid peroxide, this molecular oxygen is more exposed to the surface compared to the counterpart in arachidonol PE, so the substrate. Yeah, so the initial position was like this, and as you could see in the other lipids, it stayed like that. But for this one, it is more explored to the surface. And that confirmed the whisker model, which was suggested by other scientists, that if we have a molecular oxygen that is built into the lipids, it will be more accessible, and it will go into the membrane surface. Yes, when I had a membrane, I replicated it, and I included already this whisker model conformation in this membrane, and then I added phospholipase IPLA2 Here you can see how big this structure is. I performed simulations for two systems. This is system 1, this is system 2, and system 2 contained only the catalytic domain, and system 1 contained also ankyrin repeats. Yes, and those simulations were performed for a wild type, so regular proteins, and for a mutant, which was found in the samples of the patient with Parkinson's disease. Yes, so arginine was replaced by tryptophan. And I was studying which residues are interacting at the beginning of interactions with the lipid peroxide. Here you can see the results. Yes, and I was comparing the results, whether we have a mutant or a wild type. Yes, here you can see the final results, which we published. So this is the wild type, and this is mutant. And we could see that if we had arginine, the surface here into the catalytic site was more exposure. But if we replaced arginine with tryptophan, the binding cavity was more compact and it was more closed, I would say. Also, we have seen that we have a slightly different conformations which are available for a mutant only compared to the wild type. So those are some of the results which were published in this paper. Yeah, all analysis I was using ProD. This is a program which is very useful, and I was developing this program when I was a postdoc in Yvette's Bahar lab. Here you can see the last paper, which was published in the last year. And as you can see, it already has a lot of users, and it's a very useful tool. So if someone study biomolecules, crystal structures, and perform some alignments, it's a very useful tool to be used. So I recommend it. And here you can see the conclusion from my work. So basically, I was explaining what was did during the simulations, but we showed that PBP1 can initiate the ferroptosis when it's bind to the lipoxygenase. With nitric oxide studies, we showed that it can inhibit ferroptosis when we have a high level of nitric oxide, because it can compete with oxygen in the catalytic site. With bacterial studies, we showed that bacterial lipoxygenase, it's able to generate the same product which is generated by a human complex, even if we don't have a PBP1 and the same substrate. So the mechanism is still conserved in the structure of lipoxygenase in bacteria. And in ferrostatin studies, we showed that ferrostatin will inhibit the complex with PBP1 and not only lipoxygenase alone. And with phospholipase E-plato-beta, we showed that it's able to eliminate this ferroptotic signal. And the last work I didn't show here because it's too much results, I think. But here in this paper, we were studying 88 PDB crystal structures which are available of lipoxygenases from different species. And we were showing the differences and similarities between them. So if someone want to perform similar analysis, ProD, the program which I was showing before, it's a very nice tool to do it. And here you can see people who are involved in those projects. However, I have to say that those projects are multidisciplinary, and they're involved many different groups, not only from University of Pittsburgh, but sometimes from other university. But it's like 20 or 30 people per paper, so it's hard to include everyone, especially with photo. Yeah, and here you can see the funding, and I will be happy to take any question. And the last slide, maybe someone will be interested in participating in this conference, which we will be organizing in the next month. So thank you very much for your attention, and feel free to ask any question. I will be happy.
So if somebody has a question, you have to write this question. And I think that we got the first question. Let's have a look. This first question is the following. Are you sure that you study ferroptosis and not lipooxygenase function based on different factors?
I'm not sure whether I understand correctly the question. You mean the factors? Which factors? to be honest, whether, yeah, I would need more details about the question to be honest. Yeah, we have experimental validation, so we know what is happening in experiments, and our computational results correspond very well to those things which we can see in the experiments. So I guess that all factors are included in the experiments, or most of them, but I'm not sure what the factor is, whether this is a protein or small molecules or something else.
Okay. The second question is the following. What is the target membrane of 15-LOX PEBP1?
Okay, so we have different lipoxygenases, and they are present in many different organs and so on. So mainly it was mitochondrial, if I remember correctly. The next step would be to study the complex in the membrane. And yes, this is what I can tell right now. Yeah, but those lipoxygenases depends on which one they are present in many organs, many. Yeah.
I guess I have a similar question also because the effect your study of ferroptosis concerns the plasma membrane, right?
Yes, yes.
How it looks for intracellular membranes? I don't know, nuclear, mitochondrial, reticulum. Do you observe also the similar effects of permeabilization based on the lipid oxidation?
You know, I never, because I'm performing computational studies, I never thought about those things, to be honest. I guess my experimental collaborators would be able to answer, you know, fully on this question. Yes, but true, we have this membrane, cellular membrane, and yes, I'm not sure how to, you know, answer to your question, to be honest.
Okay, and now one issue concerning your methodology, this kind of the molecular docking based on protein docking.
Yes.
What do you need at the beginning? You need a crystal structure of two proteins, or is there a possibility to involve also the membrane in this sophisticated calculation, or it should be, let's say, water-soluble proteins? How looks this protein docking?
Yes, normally we are taking just crystal structures, which are available in a protein data bank. If not, we may perform some homology modeling. But yeah, basically we are using two crystal structures, and we are performing molecular docking just on those two structures. You can define whether if you know where is the binding site, you can define some residues where it should go, more than less. And then based on the energy of binding, you can select the complex. However, the energy of binding, it's not always is good, we may say. Sometimes the structures which has a higher energy of binding, sometimes they are better, let's say, because this docking does not include water, does not include, in this case, membrane. And if you want to add membrane into the system, you need to add it you can add it using some tools such as Charm GUI. It's very nice server, which was developed for many years, and prediction of how the protein will be inserted into the membrane, you can predict also by using some server. It's PPM, OPM server.
So this protein docking can be done also on membrane proteins, right?
Yes, however, it will not include the membrane. At least I haven't seen such servers or programs which will include that. Maybe there is something new. However, I haven't seen such.
Okay. So if you want to ask the question, please write the question in Q&A, pytania and odpowiedzi. There is a form to write the question. I have still one question from me concerning this nitric oxide, because you were using the expression competing with oxygen. So to what extent this reactivity of nitric oxide is crucial? For example, is there a possibility that carbon monoxide can do the same as nitric oxide in terms of competing with the oxygen?
Very good question. And I was testing these molecules too, to be honest. However, we didn't use those results. Yeah, that's true. That was another approach just to test CO or CO2 in those simulations and in the experiments. However, nitric oxide is common in our organism, and it has been seen to compete with oxygen, but in some other reaction. So that's why we tested.
Yeah, but carbon monoxide is also getting fashionable molecule as a donor of carbon monoxide and so on. So it may be also interesting to have a look. Still, there is, I think that we have one question more. What is the difference between binding of oxygen and nitrogen oxide to the protein on the molecular level?
They prefer similar sites, so it's hard to find differences. We were trying to find some channels which will be used, for example, only by nitric oxide or oxygen, just to mutate it and validate it. But according to the simulations and the molecular level, I can say that they are using similar binding spots. They are, let's say, competing for similar region for highly conserved residues, to be more detailed. Yeah. So it's similar place.
I do not see the question. Karolina, thanks a lot. It was very interesting.
Thank you very much.
Thanks a lot for giving responses to the questions.